So, hello everyone. Today I'll present some results of my work. This is a development of my master thesis at the University of Milan under the supervision of Simone Sarasso and Thierry New. And the aim of my project is to investigate the impact of cortical lesions on brain complexity in a whole brain computational model. In particular, here, brain complexity is understood as the spatiotemporal components of the complexity of the spatiotemporal components of the evoked activity, of the activity evoked by a perturbation, and quantify with PCI. So to do this, I use the virtual brain platform that you well know, and each node, and I use a directional connectome of available in TVB. And each node was populated with the larger Break-Sphere model. It is a voltage-based neural mass model. And then, I implement different lesions in the connectome. And following an empirical experiment with TMS EEG technique, I choose an area to be stimulated. That is in the same hemisphere of the lesion lesioned region. Then I analyzed the brain response in terms of the instantaneous firing rate of the model. I binarized this significant activity, the significant activity relative to baseline, and then I applied Lempel-Ziv complexity to the binary matrix. And I measured brain complexity via PCI. So the first result I would like to show you is about the brain response to the perturbation. And in the y-axis here you can see the delta instantaneous firing rate of some regions of the stimulated hemisphere and damaged in the lesion conditions. And you can see how in the two lesions I reported here, there is a reduction in stimulus-related oscillation after the perturbation. Then I thresholded the delta instantaneous firing rate here in order to analyze the richness of the brain response with sample entropy. That is a measure of the regularity and redundancy in a time series. So, in this particular region, that is the central-lateral prefrontal cortex, I observed a reduction in sample entropy in the two lesions. And here I did the same for each region of the connectome, above for the stimulated hemisphere and below for all the connectome. I observed a general reduction of sample entropy of the brain response in both situations, but more in the stimulated hemisphere. And interestingly, this reduction, the mean sample entropy among all regions of the stimulated hemisphere, well correlates with the mean firing rate decrease after brain injuries. Each point here represents a different lesion conditions. So the mean firing rate decrease after the lesion correlates with the mean sample entropy among different nodes. Then I analyzed the spatiotemporal complexity of the response. Here you have the spatiotemporal distribution in the brain of the evoked activity. Here you have the binarization of this significant activity. And as you can see in the control, the activity is very well differentiated, often involving different regions of the both hemispheres, and it is also temporally rich. In L2, the lesion 2 here, the activity is temporally restricted since it dissipates very rapidly, and it's also spatially restricted. In L1, you can see how the activity is spatially similar to control, but it is not very differentiated. It's very redundant. You can see the same pattern of activation over time. And it's also not very well integrated, since we have the same areas and not the both hemispheres activated. An interesting result about this is that, and that I didn't show here, is that the amount of evocativity in distant regions from the stimulated site correlates with the values of PCI. So this means that PCI is a measure of integration in this model. Overall, so I observed a reduction of PCI in the two lesions respect to control. And so this is my presentation. Thank you for the attention.